welcome to our 5th webinars of ALG-AD project. And today is my pleasure to welcome you for the theme of challenges and results after 2 years of microalgae production using digestate. And today is my pleasure also to introduce you to Darren Oatley-Radcliffe, professor at Swansea University, is chemical and bioprocess engineer, who has several years of experience in industry in oil and gas and pharmaceuticals field. And as a result, he has highly experience in the design and development of scale-up of process equipment from lab scale to the factories. In an ALG-AD project, Darren has a really important role to design and construct the PBR as a vital piece of equipment for the ALG-AD site. And so, Darren, can you please now present your talk?
Thank you, Alla. So, as Alla's just indicated, I'm Professor Darren Oatley-Radcliffe. I'm a chemical and bioprocess engineer by training, and I've got approximately a decade of experience in industry and a similar amount of experience now back at the university. And the talk today really is all about construction of the photobioreactor for Investment One, which was the British deployment site in the ALG-AD project. So as a brief synopsis, as an overview really of what I'm going to talk about, I'm just going to give some very brief notes or thoughts on the overview and the concept of the project. But then as part of that, I'll outline some of the constraints that were involved in this particular project. I'll give you an idea of the front-end design of the PBR that we eventually built. I'll talk through some of the construction activities and some of the issues and some of the efforts that we had to go to and some of the obstacles we had to overcome. And at the end, I'll show you some actual pictures of the installed photobioreactor. And at any point in time, if you want to ask some questions, fire them into the system. And if I don't answer them directly during the talk, I'll certainly take them offline and answer them afterwards for you. So as a very brief overview, in terms of the concept of what we were doing here, essentially the aim of the project was to grow algae, as I'm sure you're aware, and the idea was to create useful products from that algae. But unique, I think, in this particular project is that we were growing algae on waste materials. So the nitrogen and the phosphorus that the algae would use or consume as part of their growth would come from a waste source, and in this particular case, an anaerobic digestate. But as you can see on the screen on the bottom right-hand side here, we can effectively take waste from any sort of industrial, agricultural, domestic setting. If you put it into an AD system, the AD will break all the organic materials down. It will produce biogas, and the effluent from the AD process then is a nitrate and phosphate rich material. So we designed processes to harvest those nitrates and phosphates using membranes, and then we use that as a nutrient source for growing the algae. Now, ideally as well, we would have used an industrial flue gas to supply carbon into the system, so CO2 or CO really into the algal growth period. But in this particular case, it was too expensive to reroute the flue gases from the combined heat and power plant, which was at ALG-AD. So in the end, in this particular project, we supplied CO2 from a bottled source. Now, the idea of what we're doing is a true circular economy, essentially. So recycling, if you want, waste materials via algae, growing algae on those waste materials, and then downstream processing of those waste materials, or the algae that's produced from those waste materials, to be more precise, into products. And I'm not really concerned too much in this talk in relation to growing the algae and the downstream processing, but I'm sure Claudio in the next talk will mention some of these things. So in terms of the concept and the constraints, on the screen here you can see a very basic diagram of the technology really that we employed. So what we've got here is a light phase reactor, or a light reactor as we've called it, which is essentially a series of vertical tubes which are transparent, and they're all linked together such that they form essentially a large serpentine type reactor. We then use a tank, which is called here the dark tank on the screen, and we basically recirculate the contents of the tank through the light reactor and back to the tank again. So it basically is a batch reactor in essence that basically recirculates. But obviously with time, we could take materials out of the system and put fresh materials in, so it becomes a pseudo continuous reactor. Now, how we normally control the system, one of the major parameters is pH, and we control that by injecting carbon dioxide into the system, forms carbonic acid, and therefore lowers the pH. So as the algae grow, they consume the carbon in the system, and therefore the pH rises. So essentially, we control the reactor based on pH, but not shown on the screen. Obviously, we control the reactor in terms of dissolved oxygen content and also temperature as well, and nutrient content and things like this. Obviously, there's an off gas that comes out of this system then, and those are basically the off gases of any unused CO2 that's in the system, but also obviously the algae photosynthesize, so you get oxygen coming out of there as well. So in a previous project to the ALG-AD project, we demonstrated CO2 uptake from a wood-fired flue gas. So we demonstrated basically recycling of industrial gases. And the aim here then in this particular project was to show that we could also recycle nitrogen and phosphorus in the forms of NOx and POX. We knew that we had a reactor technology like this. We'd already built one at large pilot scale, I suppose, at the university at around about 2,000 litres. And we wanted to scale that process up. And we wanted to make a much larger reactor essentially to demonstrate that this technology was scalable. So in terms of the constraints of the ALG-AD project specifically, we already had a greenhouse in place which we could site our reactor within. So obviously we had to work within the space limitations of the current greenhouse. And also within the greenhouse itself, when we initiated the project, there was no power in there. So we had to bring power across from local buildings in order to power our system. Another major constraint obviously was our budget. And as a result of that, we couldn't recycle the CO2 flue gas that was available on site. So we used a bottle gas as a replacement, as I said. And basically, in terms of the nitrates and phosphates, we had as much of this as we could possibly imagine. The facility at Langage is quite large, so there was effectively for us an unlimited supply of nitrates and phosphates, or nutrient, if we just call it that. So what we did first up was sat down, put pen to paper, and drew up what our process was going to look like. And what you're seeing on screen here is a high-level process flow diagram. And I've circled it really into three major sections. The first section on the left in red was the anaerobic digestion effluent treatment plant. So that was sludge processing essentially to recover the nutrients that we need in order to feed the algae itself. On the top right-hand side in green, you can see the photobioreactor itself. That's obviously where we grow the algae. And then once you've grown the algae, you then need to harvest them and concentrate them. And that's what's at the bottom of the screen. So this is an overall process flow diagram of what we built and installed at the Langage facility. So in terms of the photobioreactor itself, which is really the subject of this talk, what we've got here is a P&ID, or process and instrumentation diagram, a schematic, if you will, of the actual reactor itself. So what you can see in the left-hand side here is the main tank. Down in the bottom there in green, you can see a pump, and essentially fluids then are pumped out of the tank downwards through the pump, and then they come up this blue line that you can see here into the top part of the photobioreactor at this point here. They then essentially snake their way all the way along to the end of the photobioreactor. They exit via the final tube, and then they basically return via the blue line you can see there at the top, which drops them back down into the tank. Now, what you'll also notice on this diagram is these two sort of dashed red lines highlighted, and these represent essentially the serpentine part of this reactor split in three sections. So you've got a section 1 essentially at the front, section 2 in the middle, and then section 3 at the end. And the reactor was essentially designed so that you could isolate each one of these three sections. And the idea being that when you first initiate or inoculate the reactor, you'd only use section 1, and you could return materials back to the tank from section 1 only, which means you don't need a massive inocular in order to conduct the reaction. Once you've grown the algae in section 1, you can then open up section 2 and use what's in the system to inoculate section 2. And then similarly, once you've grown the algae up in section 2, you can then open up the whole reactor and grow throughout the whole thing. And what that does is it basically facilitates cell line scale up, and also means that you don't need large inocular, typically something around about 10%, maybe 20% of the total reactor volume. You can basically scale up in stages, which helps a lot. So essentially, in terms of the construction activities, the final design was agreed and was what you just saw in the P&ID, and we produced a nominal 5,000 litre reactor with a maximum of 7,500 litres in total. And the difference in those 2,500 litres is the system was designed essentially to half fill the tank, but if we fill the tank right the way to the top, we'd have a total production volume in there of around about 7,500 litres. We had natural lighting only. That was the plan at the start. And as I said earlier, this was a similar design to designs we'd seen previous and we'd built previous, so we were confident it would work. Now, obviously, as projects change and evolve, some of the constraints change. And essentially what I said here changes on the fly, if you want. So things that were changed as we were building is we wanted to be able to supplement the natural light with artificial light. So we put in a lighting system. And also the reactor that I've just shown you there is for phototrophic growth. And we also wanted a section at the end of the reactor that could sustain mixotrophic growth. So these were additional constraints added during the project. So the design was implemented in a modular fashion. So what you're seeing on the left-hand side here is a single module of the serpentine reactor. And we built these on site at Swansea. We constructed the whole thing and then shipped down modules to the Langage site, which is just outside of Plymouth, so quite a distance. And then we connected the modules up at Plymouth. And we thought it was easier to construct it in sections like this and then assemble on site, so that we didn't have to spend lots of time at Plymouth, which is quite a distance from where our base of our team was at Swansea. We created 8 of the modules that you can see on the screen in total. There's 16 tubes per module. They're 110 millimeters by 3.5 meters tall. And the volume within each module is approximately 400 liters. And as I said, effectively then what you do is you line all these modules up, you connect them with a couple of pipes, and then you essentially create a serpentine type system. This is quite a busy picture, but it shows the construction activities essentially while we're creating the modules. So at the top left here, you can see lots of bits and pieces and fittings being constructed. So the actual fittings that you're looking at in this image are the top sections of the serpentine reactor, and individually within each one of those fittings is about 8 subcomponents, which all have to be glued, assembled and pieced together by the team. In the second slide here, you can see the essentially the acrylic tubes that we used as the photo phase for the reactor. In the bottom left-hand side, sorry, you can see the frames in pieces ready to be constructed into the overall framework. And then in the center part of the screen, so here you can see a frame being constructed. See Tom at the bottom here installing the base sections for the tubes, and you'll see that there's a central collector tube at the bottom there, and that's for draining purposes and cleaning purposes. And essentially what we had was if you want, a factory production unit, I suppose, creating these individual modules. And then what you can see on the left-hand side, essentially then, is 3 modules having been constructed. The other thing you will see here, if you've got very sharp eyes, you'll see that each module or each set of tubes has got a particular valve sticking out the top, and you can see them dotted across many of the tubes. And they are essentially air release valves. So when the system is initiated, obviously the tubes are full of air. Once the liquid starts pushing through, these valves allow the air to be released out of the system, and then the whole serpentine reactor can be flood filled with liquid with no issues in terms of getting the air out of the system. So they were an upgrade, if you want, from previous designs that we'd made, and they worked really, really well. The mixotrophic module, what we did there is we took one of the modules that we created and we took each of the tubes and essentially turned it into an airlift type reactor. So each of the tubes in the mixotrophic rig operated independently, and we just put a gas pump in there and aerated the tubes in order to create gentle mixing. So that's what you can see in the image on this particular slide. So the idea is that we grow the algae from the inocular up to a dense culture in the main reactor, and then we would harvest that and then transfer a dense culture, if you want, of the algae into this mixotrophic rig where mixotrophic growth would occur. So this is give you an indication of scale. So in total, we put 7 of the modules together to form the serpentine reactor. And what you're looking at in this particular image is the first section of the plant has been inoculated. So those of you with pretty sharp eyes will see down the bottom end, the first 3 sections here are light green. That's the inocular gone into the system. This is a standard transit van, so that'll give you an idea of the scale of this reactor. And as you can see down the right-hand side of this image, the reactor itself is approximately 25 meters long. It's about 2.5 meters wide, and it's 3 meters tall. And one of the constraints that we had within the greenhouse, those of you with very sharp eyes will see these cross beams which support the roof of the greenhouse. These were situated at 3.2 meters high. So we had to keep the reactor underneath these cross beams. Similarly, we couldn't fill the greenhouse full up with reactor, because you can see down the far end here is a set of double doors. They open up, and we needed to have forklift trucks essentially having access to the greenhouse for other purposes beyond this project. So we could only essentially line the reactor up down one side of the greenhouse. So this is a zoomed in image at the front end of the reactor. So here you can see what we call the dark tank on the left, and you can see the first module of the serpentine part of the reactor on the right hand side in the picture. And the tank is essentially connected directly then to that first part of the reactor. So here you can see the tank in the image. Here you can see the pump that's driving the process, and here you can see the tube that's bringing algae into the serpentine reactor. So it simply pumps out and goes around. And then those of you with very sharp eyes, the tank itself is being pumped out from the base underneath at roughly about this point. And then this is the return line that just brings everything back into the reactor on the recycle loop. In total, the volume in the tubes is roughly 2,800 liters. The tank itself has 5,000 liters in there. So normally we operate with an equal volume in the tubes and in the tank. So at full capacity, this system is around about 5,500 liters operating volume, something like this. Okay, so this is an image on the left-hand side there. You can see Vanessa, one of our operators, and she's basically inoculating the reactor for the first time. And what you can see in the image on the right-hand side essentially is the first 3 modules then being inoculated. You can also see in this particular image, you can see that there's lights here, and here, and another bunch here, and another bunch down the far end. And these lights are basically there to supplement the natural light of the system. So when it was first inoculated, we gave it quite a bit of light because it was quite a dark day, and that helps basically with the growth. And then this is the last slide I'm going to show really. So this is the reactor operating in normal operation mode. So at this point in time, you'll probably notice that the tubes themselves in the serpentine reactor look black. These tubes are actually have dense algal culture in them. So you can see it grows and becomes a very, very dense culture. We get up to something in the region of about 2 to 4 grams per liter in this reaction system. The other thing you will see along the bottom or along the floor, if you want, is a series of carboys. You can see a bunch of them here. And we use the carboys essentially. We keep a culture growing in them all of the time, just in case there was a problem with the reactor. That meant then we've got an on-site inocular that we could re-inoculate if need be. And obviously you can see in this particular image as well that the full serpentine reactor is up and running. You can spot that there's about half a tank full in the dark tank there. So this would be operating something like about 5,500 litres in this reaction system. So if we harvested the whole thing, in this particular case, we would get something like about 10 to 20 kilos of algae out of this particular reactor. So I'd just like to give my thanks to the team. So you can see a couple of the team there, half a dozen of us, and our names are underneath. I'm second from the left, I suppose, as you guys are looking at it. And there's a whole pile of people involved in this project that are not shown in the actual image here. And, you know, big thanks to everybody, really. This is a superb team effort to be able to produce this reactor and actually to get this whole system up and running. So at that point, I will finish and I will hand over back to Dr. Silkina.
Thank you, Darren. I just thank you very much. Really informative and nice presentation. And I would like to let you know for all participants, so you can put your comments, your questions in the chat. And after Claudio finish his presentation, we will start to do question and answer session for Claudio and Darren. So, and for now, I would like to present you Claudio Fuentes Grünewald, who is my colleague also from Swansea University. Claudio finished his degree in 2001 in Chile, and 2008 he finished his PhD in Spain, in Barcelona. And currently he's working in Swansea University in Faculty of Science and Engineering and Department of Bioscience. Claudio, algal physiologist with 25 years of experience. Half of this experience he gets from industry. So he knows academia and business activities as well. So, and his expertise is scale up of algal growth in a big photobioreactor, biochemical analysis and purification, including material external to cells. So he published more than 30 papers in microalgae biotechnology, and like I said, half of his career he spent in industry. So he focusing on commercialization of novel technique in microalgae biotechnology. So Claudio, floor is yours.
All right. Thank you very much, Alla, for the introduction. So I will share my screen.
Yes, it's perfect.
Okay. All right. So thank you very much, Alla and Clara, to organize this event. And thank you, Darren, as well, for that beautiful introduction regarding the construction of the PBR. So I'm going to talk today about the results and the challenge that we face during these 2 years of operation, which is quite amazing, because it's not easy to run a photobioreactor when you use waste. So that is why I would say that this is a quite interesting opportunity to show you our result and the challenge, of course. I work as Alla mentioned, as a research officer at Swansea University. So basically, I will use for the methodology the information that we published at the beginning of this year, 2021, and I will use the graphical after just to explain you the way that we have done this work. So basically, we use the digestate that we take from the anaerobic digestion plant, as Darren mentioned, based in Langage in Plymouth. Right, so we take this digestate and we pass through a microfiltration system, basically membrane technology, and we were able to collect the we call pure filtered digestate. So then when we have that pure filtered digestate, which is contain a high amount of ammonia and phosphate, we use that as a nutrient for the phototrophic growth. This is the reactor. So after that, when we reach the desirable density, what we have done is we use membrane concentration again, a different membrane. We have two membranes, we call the dirty membrane to treat the digestate, and the clean membrane in order to concentrate the algae and to harvest the algae. So when we reach a desirable density in the phototrophic growth, we use the membrane in order to concentrate the algae, and after that we use that concentrated algae to put in the mixotrophic module. So basically in this mixotrophic model, we add organic carbon. We tested different carbon source, and we gave them the light and the optimal condition in order to grow. And after that, we use, again, membrane filtration and centrifuge in order to get the biomass that finally, currently, actually is being used for feed trial in piglet. In this case, is cows, but currently we are doing fish trial and piglet trial with the same biomass that we produce in language. So then, as you can see, well, Darren showed a beautiful photo, and this one is a really nice photo too, because in this case, we use two different strain. We use chlorella at the beginning, and then we use Sanedesmus obliquus, which is quite robust algae. And as you can see in this picture, it was beautifully growing during several months, which is the most interesting stuff. So then in this slide, as a part of the result that we published in the paper that I mentioned, you can see the different graphs that we have obtained. For instance, in graph A on your top left, you will see the growth in an 800-liter photobioreactor in Swansea University. So we did the first test in Swansea University here, and we were able to harvest during several weeks. And at the very end, we harvest with the density around 0.8 gram per liter, and basically we have done a quite innovative approach by using this membrane technology in order to concentrate the algae. I will explain to you and I will show you why. Basically, in graph B, you see the same phototrophic curve, but after that, we use two different carbon sources, acetate and glucose, and we have run 3 experiments, basically. One with no concentrated algae with glucose and acetate, and another one with concentrated algae by using glucose. The concentration of glucose was 10 grams per liter. So as you can see, the best result were those where we use concentrated algae. So in less than 72 hours, we were able to reach over 5 grams per liter. The starting point was around 2 grams per liter, and we were able to reach over 5.5 grams per liter, which is pretty good by using chlorella in this case. And as you can see in the non-concentrated biomass, we have a good growth using glucose, but not as good as the concentrated algae. By contrast, acetate, it didn't show any growth at all. Then you can see in here, this is a zoom out of this one, but in this case it's optical density. And basically in this graph, you can see that we have a really nice growth of chlorella by using glucose. Here we have the ammonium uptake in all of them, as you can see, 52 hours in the concentrated glucose. The culture itself were able to uptake all the ammonia. Same for the carbon source. In this case, it was glucose, and the pH was relatively stable and lower in the case of the concentrated algae. Here in the bottom left, you will see in graph C that this was the number that we have obtained running for nearly a year. In this case, in the first part, chlorella growth, and then in the second part, Sanedesmus growth. We have applied exactly the same approach in Sanedesmus, but in this case, we were further and we concentrated even more using membrane technology. I have to say the membrane technology is a quite good way to concentrate the algae, because you don't damage the cell, and that's quite important, because if you use any other kind of system, like for instance centrifuge, you will damage the cell. You won't be able to use that as inoculum for the mixotrophic reaction. In this case, we concentrated up to 7 grams per liter, and then we ran, and after 48 hours we reach over 12 grams per liter, which is pretty good as well. What was the main issue? As you saw in the previous one, when we use non-concentrated algae and glucose and acetate, the problem that we have basically is that bacteria won the race against the algae, because basically, as you know, carbon source, and especially glucose and acetate, is basically sugar. So the bacteria grown a lot, and basically what we obtain at the very end is this kind of biomass, which was mainly bacteria and just a bit of microalgae biomass. But in the case of the in number C, in letter C here, you can see that in this case, by using concentrated algae, the algae won the race against the bacteria. So at the very end, we obtain a really nice color and really concentrated algae paste. So that it shows that the approach is quite a novel approach of using membrane technology and then to run a mixotrophic reactor is a good option. And the idea was to do the environmental service, I would say, in the phototrophic reactor, where we take mainly the ammonium phosphate, and then by using membrane concentration, we improve the concentration of the algae, and then we put in the mixer reactor in order to boost the growth and to get the biomass that we will need for the feed trial. Here we have more results of that. As you can see, in terms of autotrophic and mixotrophic, in terms of growth rate, they are quite similar between chlorella and cinedesmal. Also, we have a quite similar nitrogen uptake. Of course, the biomass productivity in the case of mixotrophic cinedesmal was very good and good as gooder than chlorella. And regarding biomass production, yeah, we have reached 12.5 grams per liter in the case of cinedesmal and 5.32 as an average for chlorella. Okay, so that is pretty good result, I would say. Also, we have characterized the biomass that we produce. In this case is chlorella biomass, and basically, as you can see here, when we compare with the standard F2 media, when we use in autotrophic condition, using 2.5% digestate, which was the standard concentration that we use in the photobioreactor, we were able to obtain a really nice, a really good protein concentration. Also, what we have obtained as well is, and this is quite interesting, is the amount of carotenoid that we obtain when we run in phototrophic condition by using the digestate. So as you can see here, the amount of carotenoid is way higher than the standard F2 commercial media. And of course, in mixotrophic condition, because they are consuming carbon, organic carbon, the carotenoid are a bit less than the phototrophic reaction. So basically in the figure 2C, you can see the same trend, but in this case using FTIR as a proxy to know the macromolecular composition of the biomass. Of course, we have challenges, difficult challenges, I would say, and I'm going to explain to you why. Well, first of all is the digestate composition. As you can see here in this table, this is a standard analytical report of the digestate that ALG-AD produced, and as you can see, the ammonium concentration is really high, nearly 4.2 grams per liter, which is quite high. So that was a big trouble, because probably you know, but ammonia, when there is too high, is becoming toxic for photosynthetic organisms. So that was the main challenge. Then we have some amount of copper on it, but it wasn't an issue because at the very end we dilute a lot. So basically it was okay in order to grow and have no effect on the algae. And then basically it was the total calcium content, which is pretty high as well, and that is why this digestate is quite basic in terms of pH. So we are talking about 8.5 as an average. If we normally use a pH of 7.5, so that was an issue as well. So as you can see, as a part of the issue, this is an image that you can see one of the membrane that we use at Swansea in this case, in our first attempt to to process this digestate and to filter the digestate. As you can see, it's a quite thick sludge. In this image, you see the digestate, and then we get from the permeate, we got the pure filtered digestate. But as you can imagine, it's quite dark. All right, so this is the digestate itself with all this concentration. So that is why we thought at the beginning, when we wrote the proposal, yeah, we're going to use around 20% of digestate. But the truth was that with 20%, the transmittant in the clean water was below 50%. So it was a really brownish water. So we weren't able to use, because basically algae cannot grow in there, and also the ammonium content was really toxic for them. That is why we tested in small scale different concentration, and at the very end we found that 2.5% dilution is the best in order to run the reactor, and even lower dilution, 0.5 or 1%, it keep the culture in more I would say stable condition. This was one of the first challenges. Then the second challenge, I would say, it was this, as you can see, tiny little spot of biofilm, and then after that it become in this and that. So basically, as you can see, biofilm formation was one of the issue. Why? Because when they are stressed, when the algae is stressed for abiotic parameter like pH, or even for different conditions, environmental conditions, they start to producing EPS, and the algae start to attach to the surface, internal surface of the tooth. And as you can see here, this was one of the issues that we need to sort it out. The good thing, as Darren mentioned, this is in a sectionable reactor. So basically, we can sectionize this reactor and clean each of the module without stopping the reactor itself. So basically, in this case, we stop at these 3 modules. We keep it running, these 4, and we clean this one, and then we clean the rest. So that is a really good way to tackle this biofilm formation. But the problem, when we had this biofilm formation, and that was one of the question in the chat, we need to clean the reactor. So basically, this was an operational challenge. As you can see here, we use normally a jet washing machine in order to clean the reactor and to keep it in a stable condition. And as you can see here, we move, in this case, in this picture, we move all the culture to the dark tank. We keep it running the dark tank, and basically in, I would say, 4 to 6 hours, we were able to clean everything and then restart the culture again. So it's quite handy, and that is a good point of this reactor. So basically, these were the main challenge and the main result of our work during this nearly 2 years of operation. This was my last slide, and I would like to thank, first of all, to our founders, Interact Northwest ALG-AD, of course, to Language AD Plan, because they really help us, and they were really keen to collaborate with us and to help us in all the related issues of the operational. I have to thank as well to our multicultural microalgae team. There are no faces in here, some other people, but as you can see, we are from several different countries, and it's really nice to work with all these people. And I would like to invite you, if you want to know more about microalgae, or if you want to show to your kids what is microalgae about, please download from the link below this comic. Okay, thank you very much, and I will be happy to answer your questions. Thank you.
Thank you, Claudio. Really nice presentation. Thank you very much to tell us about fantastic results and challenges as well. So it's really practical and, you know, just important to see for us as well how we progress with the results so far. Right, so now we have a Q&A session. So I really encourage you to put your questions in chat box. And I have some questions right now. So Claudio, can you start first, probably? So the question is from Hebrard. Sorry if I'm not pronounced this right. So did you encounter some issue with the carbon dioxide mass transfer?
As Dar mentioned, no, we don't have a big issue with this with the CO2, and actually no carbonate was formed during the production cycle. So basically, why? Because we normally use CO2 on demand. So basically, we set a pH in the control panel. In our case, was 7.5. And as soon as the algae start growing, they are taking the CO2, and we normally have an hysteresis of around 0.1, 0.2. So every time that the medium is becoming basic, straight away we inject the pure vaporized CO2. So it was mainly biologically uptaken by the algae. So we shouldn't or we wouldn't find any problem with that. No.
Okay. And like another question, this is like not from panel, but from myself. So how about oxygen? Some people ask usually, you know, so because algae produce oxygen, and does it affect their growth if, you know, just...
Yes, oxygen is a common issue in this reactor, and specifically in cross reactor. But for us, oxygen is a proxy of growth. So basically, when we have oxygen measured inside the reactor, and they are releasing a lot of oxygen, that means that they are growing, therefore photosynthesizing. So basically, that is a good tool in order to measure if they are growing okay or not. With the pH in combination, you can realize, all right, they are growing because they are taking a lot of CO2 and they are releasing a lot of oxygen. The good thing is that this kind of reactor, as Darren had explained, so basically we have a sump, we have a tank that we normally run in a low volume. So what happened, we have this, and we put the tube, the inlet tube to the massive tank, doing I would say, cascade or waterfall effect. So basically, all the crew to go in there, and then this waterfall effects will help to release the oxygen to the atmosphere, and then that's oxygen go down, and again we inject CO2. So it wasn't an issue.
Perfect, thank you. So another question is about the cleaning. So how you clean the vertical PBRs? I know you just explained a little bit in your presentation as well. Can you give any practical advice?
Yeah, well, the thing is that normally you need to clean at least when you run in semi-continuous mode, you need to clean at least once a month, I would say, because the algae at some point start to exhausted about their running, so they start releasing the CPAs and attach it to the internal surface of the tube. So and as I say, in this kind of design, the good thing is that it's a section of a reactor. So basically, you can move the whole water to the sump, or run just one section, and then you can clean. We clean by using a jet washer, which is pretty handy, I would say, and you can do it quite fast. Actually, we can in a standard operational condition, in less than 6 hours, we were able to clean the whole reactor and restart again. So it wasn't an issue. It was a lot of work, and I would say that Jose and Vanessa, who were the technicians that they were working in there, they were really good guys that they help us a lot, and hard work from them by cleaning and keeping this reactor in good growing conditions.
Super. Okay. I have some questions. So, Claudio, so are the vertical PBRs a batch or semi-continuous system?
It's a semi-continuous system. I mean, you can run, if you want, you can run in batch mode, but normally it's semi-continuous, because the whole idea of this is to run the reactor first to uptake the ammonia and phosphate. So we do the environmental service by bioremediation. So the idea was to run in a semi-continuous basis. That is why we were able to uptake a lot of ammonia and phosphate. And second, you have to provide biomass in I would say, in a stable and reliable way. So your biomass must be the same composition all year round. I mean, you cannot, in this case, you cannot run a batch and then stop the whole reactor and then restart again, because it takes several weeks to have this 25-meter long reactor with 5,000 liter culture fully running. So that is why, as I say again, and Darren had mentioned, the idea with this section is to start with a small inoculant and then you expand the reactor. So when you reach the steady state condition, you start harvesting normally between 20 to 30% of the volume. Yeah.
Okay. So another question from Jay. Thank you, Claudio, for the excellent presentation. Is the biochemical composition protein of biomass uniform throughout the 2 years cultivation?
Yeah, as I explained just now, yeah, that was a really good result in our case, because the biomass composition was really stable, and that it was good because at the very end, our aim in this project, and specifically for the feed trial, was to obtain the maximum protein content, because at the very end, what we have done is we have sent this biomass to our French partners in Cooper, which is a massive company of pig production, and basically in a lab next to them, they have done the hydrolyzation step, because the idea was to chop down all the proteins, you know, and get the full amino acid in there. So the total amino acid, in order to incorporate a certain percent of this biomass to the standard food. We were lucky, and our result showed that this exactly the same composition through the season as well. So not just in summer, in winter time, we had the same result, which is quite good, to be honest.
Okay, thank you, Claudio. So another question. Thank you very much for nice presentation. And I wonder, how did you deal with wastewater that you get from your own operation? Did you flush it to the municipal sewage system or recycle it somehow?
Yeah, so in this case, and that is the, I would say, the versatility of the membrane is a really versatile system, the membrane technology. So we use the membrane in order to harvest the reactor, right? Either to concentrate for the mixotrophic, or when we obtain the biomass from the mixotrophic reactor. So when you pass the culture through the membrane, we obtain a crystal clear water, all right? Because this is an actually standard microfiltration, 0.2 micron, you can obtain actually, it's a kind of mechanical sterilization. So technically, you can reuse that water for several cycles. In our case, sometimes we did that. Some other time we flush it away to the municipal water. The good thing is that all the numbers in terms of ammonia and heavy metal were below the level, which is good. So we have demonstrated that we can use this as a tool, or microalgae can be a bioremediation tool, right? You can reuse the water, I would say maximum 3 to 4 cycles, no more than that, because otherwise then it's too much organic material on it. It's not going to be good. So every now and then you need to feed the reactor with new fresh tap water, in this case, because it was freshwater algae.
Okay. Right, another 2 questions, Claudia, and then we'll go to Darren's.
Yeah.
So, sorry. Right, so instead of depending on single species, why can't we use a mixture of algal species, which makes the process stronger?
Yes, could be stronger, but I have my concern about the using consortia, right? Consortia is a, and we tested at the beginning, right? So actually we went to language, we collect sample in a leak of the anaerobic digestion tank, and we used that one, and it was a mixture of male cyanobacteria, diatom, and some chlorophytes in there. The problem that you have with there, yes, you can update and could be really good a tool in order to bioremediate, right? But it's not good enough in order to get a reliable biomass composition, because as probably you know, some dinosaurs will grow better in the spring, some cyanobacteria will grow better in summer, and some green algae will grow better in winter time. So then your biomass composition change a lot. And if your aim is to produce any specific metabolite, protein, fatty acid, or even pigment, then it's not reliable, it's not constant, it's not stable during the whole year. So that is the main issue. So summary, if you want to use just bioremediate, yes, for sure, go for consortium. If you want to bioremediate and produce good quality biomass, you have to use single species.
Okay, super. And very last question. I still have some, but I think so we can answer everything. Anyway, so concerning the nitrogen uptake is calculated on the basis of the biological consumption by algae only, or with the stripping part from the ammonia as well? It's a good question.
Yeah, this is a really good question, because we understand that most of the ammonia is being uptaken by the algae. But to be honest with you, we haven't done that, but we're gonna do probably. We're gonna measure the ammonia that is released to the atmosphere. So that is a balance that we need to try to understand, because yes, we can uptake the ammonia, and the algae, as you saw, they were really green, and actually the chlorophyll content was really high. So basically it was a lot of nitrogen. We got the elemental composition of the algae, we got the data of the CNH, and we're going to publish this as well. But definitely it would be good if we would be able to put kind of an ammonia recorder, in the sample, in order to know what is the amount of ammonia released into the atmosphere. But this is a work that we need to do, and potentially we want to sort it out during the next couple of months.
Thank you, Claudio. Right, and very, very last question. Do you know how the final product of the process is integrated into animal food?
Well, if I understand well, we are currently doing the experiment. So in France, they are using pig legs with our biomass, and with the other partner biomass from Belgium and from France as well. They have grew other species. In the case of Belgium, consortia. In the case of France, Translocutron, and they are they are testing this piglet with our biomass. In our case at Swansea University, we are tested in feed for fish, and we are currently doing that experiment. So yes, then the issue is the legal framework. All right, so technically it's feasible to do it. It's not a problem for us. Economically, yes, let's see. If we equalize the volume of a big plant, AD plant, and we have the same volume in a microalgae plant, potentially we can do it. We can be able to be a profitable business. But in terms of the legal framework, the politicians, stakeholder, and the rest of the people, they're a bit behind, because technically we couldn't incorporate this to the fish trial. But anyway, I think they're doing their work, and potentially in the near future we will be able to use this either for fish, or basically if you get the biomass, you can produce, for instance, biostimulant as well. So at the very end, you will use the biomass. You will valorize the biomass.
Super. Thank you very much, Claudio. And now I would like to invite Darren. Darren, you here?
I am indeed.
Yes. So I would like to, so several questions for you as well. So one of the questions related to the large-scale system. So in large-scale system industry require continuous system. How fit is your system for the continuous operation if it will be demanded?
Yeah, I mean, it's another great question. It doesn't really matter whether you've got a batch or a continuous system. In reality, it's how often can you harvest that, and how do you deal with it. So for example, I come from pharmaceuticals, and there's lots of different examples of batch production systems that produce quality product on a continuous basis. And you do this in the specific terms with the algae, is we would harvest multiple systems several times a week in order to produce a continuous supply into the downstream processing. So it becomes more of a case of scheduling rather than a difficulty. But in terms of running the bioreactor as we saw it today, yes, this can run in a pseudo-continuous fashion. And we did that basically for a year with, I wouldn't say without issue. We had issues from time to time. We had to clean it and things like this. But in essence, yes, it works. And certainly the demonstrator was good enough to suggest that if you scaled it up to like a major industrial plant, yes, you could run that thing as a pseudo-continuous system if need be.
Super, thank you. Darren, so how about the recycling of water after the process as well? So not to put, do you have any ideas how we can improve the system?
Yes, it was a great question actually that somebody asked in relation to the water, and I think Claudio answered it for the most part, in that we use membrane processes to harvest the algae, but also the waste product then is the water. And I think from memory, I'd have to check back in my notes, but we were recycling the water, I think 4 times before we discharged it. And the problem that you will have is, particularly because our nutrients are coming from a waste source, is you'll start accumulating things in that water over time. So every now and again, you've got to have a purge just to get clean water into the system. But if you wanted to invest a bit more time and effort, and generally that means money, there's no reason why you couldn't introduce like a nanofiltration kind of process and really clean that water up to ultra pure standards. So most people, I think, on the call will understand seawater desalination processes to produce drinking water. You can do exactly the same thing with the effluent water from the membranes of this plant. You essentially in a desalination plant, as an example, you would normally do a front end cleanup of the seawater. That's essentially what we're doing in this algal process, which produces then a clean water supply, or a cleaner water supply, that can go into the RO process and produce the ultra clean water. So yes, in essence, we are doing the pre-treatment for an RO plant naturally in this process. So if we bolted on the RO plant, in reality, there's no reason why you couldn't recycle 80–90% of this water indefinitely, and then have a very, very small purge to get rid of the heavy metals and such that may accumulate in there.
Yeah. And secondary metabolite from culture. So
yeah. I mean, I said the heavy metals, but it would include everything really.
Yeah.
You would need to purge at some point.
Yeah. And I think so this process will be make LCA improvement and so on. So
yeah, absolutely.
Let's envisage this for future projects. But another question from Jay. So thank you, Darren, for the excellent presentation. Is there any technological solution to eliminate the biofouling in the vertical reactors?
In essence, yes and no. I'll explain. If you've got very good control of your reactor, then you limit biofouling straight away. So if you keep the algae nice and happy, which means you you keep very tight control of pH, you keep very tight control of temperature. If you do this, then you'll keep the algae nice and happy, and therefore you'll get less natural fouling to start with. So that's the first thing. And when we built this particular reactor, it was the biggest reactor of its type that we built ever. And what we found is because we had quite a large serpentine part to the reactor, it meant that, for example, on a very sunny day, you could get quite a large temperature rise along the length of the serpentine. We had temperature control in the system, but what it meant was at the front of the serpentine to the back of the serpentine part of the reactor, you might have a 2 to 3 to 4 degree temperature rise. So I think if you put intermittent heat exchange in there so that you kept the whole reactor at roughly a constant temperature, you would improve that situation dramatically. Similarly, gas exchange. So we, in this particular reactor, we did all of our gas exchange in terms of introducing CO2 where the pump was. That means then, as you go along the length of the serpentine reactor, you get a pH change, because obviously the algae consumes that CO2 as you're running along the length of the photo section of the reactor. So what we could do there is have multiple injection points for CO2 so that we maintain the pH more consistent throughout the whole volume internally of the reactor. And the last thing there, obviously, as well, is gas exchange in terms of getting the oxygen out. So we basically remove oxygen from that system, all in the dark tank part of it. And our purge basically is in the tank itself. but we could in principle have disengagement ports at different parts of the reactor to again keep the dissolved oxygen content lower, or more consistent, I should say, throughout. So yes, there are fundamental design improvements that can be made for this. And we're basically, each time we build one of these reactors, we're learning a little bit more each time. So they will effectively come into future designs essentially. Will we ever totally eliminate biofouling? I don't think so. If I'm being honest, this is an organism. We can't tell it what to do without it giving us a bit of reaction, I suppose, at some point. But we can reduce it significantly, I think.
Super, thank you. Thank you very much, Darren. I think so we come out to our questions, and some of them you answer as well in writing. So thank you very much for the first questions. So I would like to finish our event today. So if you can have few minutes to answer our survey, so please, it will take like 1 minute or 2 minutes of your time. It will be really useful for us for our future event planning and organization. And I would like to thank you, all of you, for the active participation and really nice questions, and really fantastic presentation of the speakers, and Clara as well, who organized this meeting today as well. And I would like to also invite you for a very interesting event, what will be 25th of May. We will do an exhibition of ALG-AD project. And this exhibition is supposed to be not online, but real exhibition, but due conditions of COVID right now, we have to change the format of this event, but we will be making this in REMO platform. So we'll see fantastic opportunity to see all results from the project, and also meet all scientists from the project for networking, for exchange, for listening of talks and presentation. And so please follow our social media. We'll see registration links to this event. This will be free as well. And like, yeah, in the chat box as well, yeah, Clara sending a link, so you can do is already right now. It will be really interactive, really interesting, and I really welcome you to this event as well to join us. So thank you very much again, and thank you to reply for the questions as well on our survey. And I wish you a very good afternoon and very good evening, and see you soon.